Hi, this is Mr. Wartsky again, and this is the second screencast of the protein synthesis series. This is the last slide that we talked about in the production of mRNA. And I said we were going to start off talking about the code. Now, in order to talk about the code, we have to learn about a codon. DNA codes for proteins. Proteins are made up of amino acids, so DNA codes for the amino acid sequence. We got that. The code for amino acid sequence is passed to the mRNA. Hopefully that makes sense. The code for an amino acid is referred to as a codon. And a codon are three adjacent mRNA bases. So the three bases that are together equals one amino acid. So there are 64 possible codons. Like how? Well, if there are three bases, this is the first base, second base, third base. The first base has four possibilities, A, C, U, or G. The second, A, C, U, or G. A, C, U, or G. So there's 4 times 4 times 4 is equal to 64. Of those 64 codons, 3 are stop codons. And those are UAA, UAG, or UGA, or the University of Georgia. So we have UG and UGA, or UAA, UAG, or UGA. There's a start codon. That's going to start protein synthesis, and that is AUG, which codes for FMET. Well, what do you mean? Well, let's take a look at AUG. So right here we have the first base. I don't like this one here. Is up here. The second base is here. The third base is here. So AUG, line up the first base with the second base. Now see, they did this wrong. The first base is here, second base, third base, which is what it should be. A, right, line up with U, and G here is methionine, which is start. Let's give another one. Let's do the stop one, which is UGA, right? This is U, this is G, and A is stop. Let's do another one. Sure, let's do GAC. G is over here, the first. A is over here, so the G and the A line up here, and GAC is here, and that is aspartic acid. That's how we use a codon table. They also have this codon wheel. Here, we start off in the middle, like A, right? Then we have U, and then we have G, which is MET. Or we can say G, or was GAC we said? G is over here, A is over here, C is over here, aspartic acid. This is actually probably pretty easy to use, but they always give you a codon table. And we're going to practice that in class. Don't worry about that. A ribosome will bind to the mRNA molecule. Now, a ribosome has a 50S, which is a large subunit, and a 30S. Now, the S stands for Svedberg unit, and that's how fast something sinks. So the large subunit is going to sink faster than the small subunit. There are three sites, the EPA, exit, peptide, amino acid. So this is a peptide binding site, amino acid binding site, right? EPA, easy to remember the order right there. And that is the ribosome. Now the subunits come apart, they separate. The tRNA is what will bring the amino acid to the mRNA and join in the ribosome. There are 20 different amino acids carried by 31 tRNA molecules for 61 codons. Now the tRNA, they always tell me tRNA looks like a 3-leaf clover, and we're going to show you in a picture in a second. So we have the tRNA, they have the acceptor arm, the CAA end that has the amino acid. That's the 5' end. There's the D arm, not the DARM, the D arm, because it has a special base called dihydrouridine. The anti-codon arm is what's going to bind to the codon. It's probably one of the more important arms. The variable loop, there's two classes. There's a small one and a larger one. There's a T-Cy-C arm. The Cy stands for the special base pseudouridine. And there's an enzyme, amino-acyl tRNA synthase, which will add the amino acid to the specific tRNA molecule. So there's 20 of them, one for each amino acid. We have a tRNA diagram here. We have the 5' end here, 3' end up here. The acceptor arm, so the amino acid is added. Right, we have the D arm here, We have the anti-codon here. This is the T side C arm here. The variable loop is right here. And this is only what? 1, 2, 3, 4. So it's the small loop. So in translation, these are the steps. So write these things down. First, the 30S will bind to the 5' end of the mRNA. Finds that by looking for the special base, the methylguanine. The 50S will bind to the 30S, and it forms something like an mRNA sandwich. Now, the P site is over the start codon. The correct tRNA molecule, or anticodon, binds to the mRNA codon in the P site. So it enters the P site. The correct tRNA anticodon binds to the mRNA codon in the A site. So now the two sites are filled. There's a synthesis reaction between the two amino acids, and a peptide bond is formed. Hit pause if you need to. We have the 30S here. We have the 50S here. Forms a sandwich. The correct tRNA anticodon binds to the codon here, right? Brings with it an amino acid. The correct anticodon binds to the A site, mRNA in the A site. We have a synthesis reaction. Beautiful. Now what happens? Well, the ribosome will move over one codon to the right. And as it does that, this right here will enter the E site and then exit. But that will open up the A site so another tRNA can come in and join. So the ribosome moves down one codon in step 6. That's called translocation. The energy supply is GTP, and the enzyme is elongation factor P. 7, the tRNA leaves the E site, leaves the amino acid behind, and you repeat steps 4 through 6 until a stop codon is reached. Should be 4 through 7, until a stop codon is reached. And we have something that looks like that. So the ribosome is moving to the right. The 30S is on top, 50S on the bottom. It doesn't really matter. Right? The next one comes here and here, here, and we have a growing chain of amino acids bonded together. That is translation. Now, there is such a thing as a wobble effect because we have a linear mRNA, but we have a curved tRNA. So there is some flexibility in the binding of the first and the third part of the anticodon to the first and third of the codon. Yeah, so if you have a U on the anticodon base, it can bind to either A or G. If there is a G right here, you can either bind to C or U. Same with the other side here, right? G can bind with U. So they believe that's how the 31 tRNA molecules bind to the 61 codons. Polyribosomes, when you have a ribosome, makes an average size polypeptide in less than a minute, but more than one ribosome can attach to the mRNA at the same time. So you can make many different proteins at the same time. That's called efficiency, ladies and gentlemen. Now, the endoplasmic reticulum. There are two types of ribosomes in eukaryotes. They're free and bound. All the ribosomes start off as free. If a polypeptide needs to be modified, it goes to the endoplasmic reticulum. And how does it know to go there? There is a signal recognition particle. That means there's a section of the mRNA, the first 20 amino acids is the SRP, and that tells the ribosome to attach to the endoplasmic reticulum. So the polypeptide goes into the tube of the endoplasmic reticulum. It's called the cisternal space, and that is modified there, and then it will go to the Golgi complex. Now, if the ribosome stays unbound, that polypeptide is going to have a 4 amino acid sequence at the beginning of it, and that acts as a zip code. It tells the cell where this polypeptide needs to go. That's freaking amazing. So here we go. The ribosome You see the signal recognition particle at the beginning here? Tells the ribosome to attach. It goes into the cisternal space, and then the polypeptide is modified. So how is it modified? Well, in the endoplasmic reticulum, carbohydrates can be added, lipids can be added, amino acids can be cut off like F-met. The 3D shape can change. Now, if the polypeptide is going to be secreted, it has to go to the Golgi complex so it can be modified and packaged for export. Now, the Golgi complex, there are two phases. It moves from the cis phase, where it accepts the polypeptide from the endoplasmic reticulum. It moves to the trans phase, where it's modified, and then it is shipped out of the cell and secreted. We have a picture of the polypeptide going from the endoplasmic reticulum to the Golgi complex. Amazing, isn't it? So if it stays, I told you before, it has a 4 amino acid sequence at the beginning of the polypeptide, which tells the protein where to go. Acts like its zip code. So we're going to stop there for translation. Hope everything made sense. We're going to wrap this puppy up with mutations in the next screencast. Have a great night.